So welcome to everybody to this seminar. Thank you, Professor Lustig, for being with us. It's a pleasure to have you with us. Just a few words, but it's redundant to introduce you because everybody here knows about you and your career, but I have to tell some things about you. So Professor Lustig is
none of them are true. None of them are true.
No, I guess some of them would be. At least we have some proof. So Professor Lustig is an Emeritus Professor of Pediatrics at the Division of Endocrinology at the University of California, San Francisco. He is a neuroendocrinologist, and he has studied the regulation of the energy balance by the central nervous system. His research and his clinical practice has focused on the childhood obesity and diabetes, and he had some interesting work that I would like to discuss afterwards, perhaps about the prenatal obesity that is related to the imprinting of the mother to the offspring. For instance, just to mention some of his work, and he has, of course, a solid track record of publications and reviews that support his career. He has also an interesting position about the sugar and the fructose and the effect on nutrition. So really, I'm personally looking forward to hear your seminar, and I have a lot of questions at the end. So welcome to our yellow seminar. Thank you for accepting our invitation, and now the time is yours.
Well, thank you so much, Dr. Rosso, and thank all of you. Buongiorno, buonasera, whatever it is over there. It's 4 p.m. And for those of you who are Jewish, Chag Sameach. I will be cooking the rest of the day a no sugar, no gluten Passover. So first of all, thank you very much for the invitation. I am very interested in the liver, even though I'm an endocrinologist, because the liver is an endocrine organ. It makes hormones, it releases hormones, and so we endocrinologists want to steal it from you. And you will see why the liver is the central factor involved in this phenomenon we call metabolic syndrome, having nothing to do with obesity, but through its manifestation of fatty liver disease. How does fatty liver disease ultimately impact on metabolic syndrome and vice versa? And that's what we're going to talk about today. So first, this is a paper that came out now 23 years ago. Okay, it was in Nature. It talked about the data from 2000. And at that point in time, there were 151 million people walking the earth with diabetes. And what was projected was that by the year 2010, so 1 decade later, that number would increase up to 221 million. That would be a 46% increase, or as the actuaries do it, an annual inflation rate of 3.88%. This is not what we saw. What we actually saw was that instead of 221, it was 285 million. That's an annualized inflation rate of 6.55%. By 2014, with all of the gym memberships and all of the diet sweeteners and all of the attention that had been placed on obesity as the predominant problem of the 21st century, we were up to 422 million. By 2021, we were up to 537 million. Now what is projected for 2030 is 568 million. And by 2050, we will be up to 785 million people with diabetes, and we cannot afford it. We couldn't afford it before. We certainly can't afford it now. Now, the problem in trying to deal with this is that we are trying to solve the wrong problem. And the reason for the wrong problem is myriad. It's, baked into the cake. But for us doctors to be able to solve the problem, to fix the problem, we have to understand what the myths around this classification, this structure that we have unfortunately contributed to. We have to be able to take it down. And so I'm going to try to do that in 10 minutes. Okay, I don't know if I can, but I will do my best. There are 3 myths that have contributed to this idea. The first is that obesity is the problem. Obesity is the cause of chronic disease, that obesity is the cause of NAFLD, that obesity is the cause of type 2 diabetes. Well, let's take a look at that. Here we're looking at data from 2015 to 2020, and you will notice that the increase in type 2 diabetes in the overweight have only gone up by 2.1%, 2.1 fold, sorry, 2.1 fold. Whereas within lean adults, it's gone up 17.8 fold. This is crazy. The lean people are getting more diabetes now than the fat people. If it's obesity, how does that get explained? And this is true for children as well. Children get type 2 diabetes also. And you can see here from 2001 to 2017, it doesn't matter if it's, young adolescents or older adolescents, and it doesn't matter what the demographics are. In every single case, the type 2 diabetes prevalence has gone up in children. Well, you could say that could be because they're obese. Well, this meta-analysis sorts pediatric type 2 by weight. And it doesn't matter if you look at all studies or stratified by race. The bottom line is that 25% of the type 2 diabetes in children are in children of normal weight. So normal weight people get metabolic syndrome too. Normal weight people get fatty liver disease too. And the question is, if obesity is the cause, then how do you explain that? Well, here's the problem. Here we have a Venn diagram of the entire US adult population, 260 million people, 42.4% obese, 110 million, 56.7% normal weight, 150 million. So BMI over 30, BMI under 30, okay? Mutually exclusive circles. Now, the standard thought of the doctors and the nutritionists and the Institute of Medicine and the NIH and the White House and Congress and the food industry is the following. 80% of these 42.4%, 80% of these obese people, these 86 million people here, they're sick. They're fat and they're sick, and they're sick because they're fat. And if we could only just get these people here to diet and exercise, we could solve the problem. That is not true. And the reason it's not true is because 60% of the normal weight people have the exact same diseases as do the obese. Normal weight people get type 2 diabetes, hypertension, dyslipidemia, cardiovascular disease, cancer, fatty liver disease, polycystic ovarian disease also. And the reason is because these 8 diseases are the diseases of mitochondrial dysfunction, and you don't have to be obese to have mitochondrial dysfunction. Anyone with mitochondrial dysfunction is sick, including normal weight people. And it turns out there are actually more thin sick people than there are fat sick people, and they're calling the fat sick people the problem. How do we know this? Because we can image them. So here we have 2 equally weighted people, CTs through the abdomen of each of these 2, and you all are, expert at looking at this. One is healthy, one is sick. And of course, the one who is sick is B, because he's got intra-abdominal fat. This guy has subcutaneous fat, which is cosmetically undesirable, but metabolically inert. So ultimately, obesity is not the cause of chronic disease because thin people get sick too. Second myth, chronic disease is due to the fat you measure on the scale. Well, let's take a look at this. Here we have a cohort of patients from Ecuador, and they are known as the Little Women of Loja. They were written up in the New England Journal of Medicine back in 1990. And here they were back in the late 80s with their doctor, and here they are much more recently, like in the mid-2010s, with their doctor. And what you will notice is they were obese before and they're obese now. Okay, that's what you notice. But when you look at whether or not any of them have diabetes, their relatives do. Their relatives have cancer. They do not. This is a founder effect of the growth hormone receptor. In other words, they are Laron dwarfs in the middle of Ecuador, okay, a founder effect. And turns out they don't get cancer or diabetes. Oh, they have obesity for sure, but they don't get diabetes and they don't get cancer. And they also don't get fatty liver disease. Ha ha. Now, if you look at their fasting insulin compared to their relatives, it's low. They're obese, but they have low fasting insulin. Take a look at their curves. Their glucose tolerance test is the same as the relatives, but look at the insulin. Okay, here's the controls, their relatives, and here they are. They are insulin sensitive. Okay, and you can see their percent body fat is way up. Their fasting insulin is way down. Their HOMA is low. Their LDL is high, but their HDL is higher, and their triglyceride is low. So if you've got a high HDL and a low triglyceride, you are insulin sensitive. These are obese people who are insulin sensitive and don't get fatty liver disease. So clearly, obesity is not the driver of this. Here's another disease that's the opposite. This is lipodystrophy, well known to most of you, I'm sure. Okay, these patients have too little fat. And because they have too little fat, the fat that they do have ends up in places it shouldn't, like, for instance, the liver, causing severe acanthosis nigricans and severe metabolic syndrome with massive type 2 diabetes. And the reason for this is because there are 3 fat depots and they are not the same. The first fat depot, of course, is subcutaneous fat. Now, what you learned in medical school, what I learned in medical school was you get fat. Because you get fat, you become insulin resistant. You have increased portal FFA cytokines, which reach the liver. The liver responds and increases hepatic gluconeogenesis, hepatic glucose output. That then causes the beta cells to increase their amount of insulin. And so you end up with a vicious cycle of increased fat, increased glucose, and therefore increased insulin. This is the standard model of insulin resistance. And then the muscle comes along for the ride. This is not true. What I just showed you is not true, and I'll show you why at the end. Now, how is it not true? Well, take a look at this curve. Here we have BMI compared to insulin sensitivity. And what you can see is you can see the hyperbolic relationship between the two, as Richard Bergman and many, many other people have noted. And that is true. There is a hyperbolic relationship. But within that hyperbolic relationship, there are half the people who are insulin sensitive at any given BMI and half the people who are insulin resistant at any given BMI. So the curve is hyperbolic, but the individual data points may be all over the place. And this translates into morbidity. So here we have insulin sensitive patients versus insulin resistant patients for diabetes. Here we have insulin sensitive versus insulin resistant patients for heart disease. And you'll notice that for each, yes, as the BMI goes up, the prevalence rate goes up. True. But take a look at the insulin sensitive versus the insulin resistant. At any given BMI, the insulin sensitive have very little disease. And if you look at the Kaplan-Meier curve, it's not the obesity that translates into an improved Kaplan-Meier curve. It's the insulin sensitivity. Those that are insulin resistant, whether they're obese or not, are going to die earlier. So the sub-Q fat is not the problem. Second fat depot, the visceral fat. Now, the visceral fat is a problem, no question about that. But the visceral fat has a dichotomy, has a paradox. And that paradox is that when the peripheral sympathetic nervous system, the autonomic nervous system, activates adipocytes in the visceral adipose, You should get lipolysis. You should end up burning fat and losing weight. This is what exercise does. However, what is actually happening is that the visceral fat is under the control of the autonomic nervous system in chronic stress. So yes, the norepinephrine will bind to its beta-3 adrenergic receptor to induce lipolysis, but under chronic stress, the intermediolateral cell column of the spinal cord that sends the autonomic fibers to the adipose tissue, it releases a second factor called neuropeptide Y. And that NPY binds to the Y2 receptor and actually blocks beta-3 adrenergic receptor activation. It increases preadipocyte proliferation. And what it ultimately leads to is lipid storage and angiogenesis. And that is what drives the fat. So the acute stress will cause weight loss, especially from the visceral fat compartment, but the chronic stress will cause weight gain. So visceral fat is not actually a phenomenon of diet. It is a phenomenon of stress. Okay. And normal weight people get stressed too. And finally, the third, and the one we're going to talk most about, the liver fat. Okay. So of course, here's the histology of normal liver, all well known to all of you, the sinusoids, the biocanaliculi, the Kupffer cells, etc. All good. And here we have fatty liver disease. Okay. And you can see the fat vacuoles, you can see the infiltration of the macrophages, and you can see the start of scarring here. The question is, if you just saw this under the microscope, could you tell if this was alcoholic fatty liver disease or non-alcoholic fatty liver disease? You couldn't tell the difference. They look exactly the same. Okay, and that's the point. They are the same. Okay, non-alcoholic fatty liver disease, as you know, is a silent killer, known, well known to you. Here in the United States, non-alcoholic fatty liver disease has overtaken hepatitis C as the cause of liver transplants. So this is a huge problem. Now, you would say, well, every patient with fatty liver that I've taken care of is obese, and you would be wrong. Okay, so here we have a bunch of different studies, including Italy. Okay, and yes, the NAFLD prevalence in the overweight is much higher, but there is a signal even in the normal weight population. Okay, and if you look here, we have BMI bind here, going up. And here's the prevalence of non-alcoholic fatty liver disease. And you can see a very clear signal in the overweight category, even in the normal weight category, clearly less than the obese. I agree with that. But take a look over here on the right side. No diabetes versus diabetes. Okay. And again, this has nothing to do with BMI, because look at the signal, even in the normal weight who have diabetes. The amount of fatty liver is huge. So this is not related to obesity. So this is the image that I want you to take away from today's talk, this next set of slides. Here we have an MRI fat fraction map of an obese person. You can see the sub-Q fat very clearly, but take a look at the liver fat, 2.6%, nothing. Okay, metabolically healthy obese. This guy might outlive you. Now take a look at this guy. Notice obesity, but take a look at his liver, 24% liver fat. Metabolic syndrome, fatty liver disease, this guy's going to die. Now take a look at this guy, normal weight. Notice very little in the way of subcutaneous fat here, but take a look at his liver, 23% liver fat. Thin sick, fat healthy. Okay? This is the important message to get out regarding this. Everyone is at risk. And it doesn't matter if you're old or young, here adolescent NAFLD, you are at risk for type 2 diabetes and therefore premature death. Okay? 2 points, the relative risk ratio is 2.6 if you have fatty liver as a child. Now, the question, obviously, is this is a new disease. This was never seen before 1980. Where did the fat come from in the liver? Since that's the thing that has to go away. Well, this was work from Fabrini et al. At Sam Klein's group at WashU St. Louis. And what they saw was that when you did MRIs and quantitated the amount of visceral versus subcutaneous fat, and they binned them, and then they did a multivariate linear regression analysis, holding either visceral fat constant and looked at the liver fat here in the first two columns, or held liver fat constant and looked at the visceral fat in the second two columns. What they noticed was that when you held the liver fat constant, the visceral fat explained none of the metabolic change. What did was when you held the visceral fat constant, the liver fat explained all of the metabolic change. It's the liver fat that makes the difference. So the question is, where's the liver fat coming from? And in this slide, you can see over here in gray, non-systemic fatty acids. Now, we know where systemic fatty acids come from. They come from diet, or they come from lipolysis of adipose tissue. But non-systemic, those are fats that are made right in the liver through the process of de novo lipogenesis. So you have heard, I'm sure, as being a hepatologist, of the two-hit theory for fatty liver disease. Okay, I am here to tell you that is incorrect. It is not the two-hit theory. It is the three-hit theory. And one of those hits is metabolic. And you'll see in a minute why. So, as you know, the American Liver Foundation and the NIH, in its infinite wisdom, has decided to rebrand non-alcoholic fatty liver disease to now be called metabolically associated steatotic liver disease. Does this make sense? Well, let's take a look at that. So we have to go through three hits, and I will explain each one, and all of them will be known to you, but let's string them together so that they make sense. The first hit, mitochondrial dysfunction. Okay, if your mitochondria are working, then you're burning energy in the liver. You're not storing it. You're not making fat. So inherently, there has to be mitochondrial dysfunction to lead to fatty liver. Okay, and the question is, through what mechanism? Well, you know, we pediatricians know this well. Perhaps you do too. Okay, in America, I took care of, when I was a resident, 45 years ago, I used to take care of kids with Rye syndrome all the time. Okay, and they were horrible, and they didn't get better, and a lot of them died. Right? And the question, of course, is, what caused all this fatty liver and Rye syndrome? Well, we learned that aspirin is the driver, and the reason is because aspirin is a mitochondrial toxin. So when you're sick and you give little kids aspirin, okay, you basically make their mitochondria stop working. And when their mitochondria stop working, as you can see here, the salicylate level goes up, and you can see the palmitate oxidation goes down. Okay, when that happens, you end up generating hepatic lipid. Okay, so mitochondrial dysfunction of any cause will ultimately lead to the generation of fat, because you're not burning it, you're going to store it instead. Okay, what are you storing? Well, I mean, what are you inhibiting? Well, for Rye syndrome, turns out aspirin inhibits this enzyme right here, long chain hydroxyacyl CoA dehydrogenase, also known as ACAD-L. ACAD-L. Please remember ACAD-L. We're going to come back to it in just a minute. All right. Now, what could be causing mitochondrial dysfunction in NAFLD? Obviously, it's not Rye syndrome, which is acute. What could be causing it chronically? And this is our work at UCSF, which was written up in the New York Times Magazine back in 2011 by Gary Taubes, Is Sugar Toxic? And then we wrote this comment in Nature back in 2012, The Toxic Truth About Sugar. And we meant it. We meant it because sugar is, in fact, a mitochondrial toxin. So, as you know, of course, sucrose, table sugar, is composed of two molecules that are bound together, glucose and fructose. The glucose is not the problem. The glucose will enter the liver, get converted to glucose 6-phosphate through glucokinase. That virtually all of it will end up as glycogen. Okay, that's how the liver wants to store energy, is glycogen. Very little of that glucose 6-phosphate will make its way down to pyruvate through the Embden-Meyerhof pathway. That pyruvate will then enter the mitochondria, be converted to acetyl-CoA, and will participate in the Krebs cycle, generating ATP and carbon dioxide. Old news to all of you. Now, if you overload your mitochondria with pyruvate, and the Krebs cycle's Vmax is exceeded, then the Krebs cycle will throw off the excess as citrate. The citrate will then depart, will leave the mitochondria through a process known as the citrate shuttle, and then that citrate will be substrate for the 3 enzymes of de novo lipogenesis. That is acetyl-CoA carboxylase, ATP synthase, acetyl-CoA carboxylase 1, and fatty acid synthase, those 3 enzymes, which are, by the way, driven by AKT, which is driven by insulin. So when insulin goes up, de novo lipogenesis goes up, as long as there's substrate. So if you give excess glucose, you will end up with increased free fatty acid formation in the liver, de novo lipogenesis, which will then get packaged into triglyceride and hopefully exported out of the liver to take up residence in the peripheral adipocyte. So this is how starch makes you get fat. But it doesn't make fatty liver because the majority of that glucose goes to glycogen. Fructose, on the other hand, is a different story entirely, because fructose is not glucose. Fructose is not under insulin regulation. Fructose gets turned into fructose 1-phosphate. That causes a loss of ATP. That leads to uric acid. That uric acid will be the endogenous inhibitor of endothelial nitric oxide synthase. That is your endogenous blood pressure lowerer. And so your blood pressure goes up. There's your hypertension. In addition, the fructose 1-phosphate will come straight down to pyruvate, all of it, the 100% of the load, because the liver doesn't turn it into glycogen. That pyruvate will then enter the mitochondria, overwhelm the mitochondria's TCA cycle Vmax. Now you've got a lot of citrate instead of a little citrate. Now you got a lot. And now that citrate will be acted on by those 3 enzymes to generate triglyceride. Some of the triglyceride will make it out and go to the adipose tissue. Some of the triglyceride won't, and it will precipitate as a lipid droplet. There's your fatty liver disease here. In addition, fructose 1-phosphate through C-Jun N-terminal kinase 1 will actually serine phosphorylate IRS1, generating hepatic insulin resistance. So now you're insulin resistant as well. The triglyceride will lead to adiposity or dyslipidemia. And of course, the insulin resistance will lead to pancreatic hyperfunction, which in the long term will lead to diabetes. In addition, we have learned that insulin blocks leptin signaling. And if you block leptin signaling, your brain thinks you're starving, in which case you're going to eat more. And so you've turned a negative feedback pathway into a positive feedback pathway through this insulin resistance, leptin resistance phenomenon. So how do we know that this is true? Well, because we can see it, all right? This is one of the studies that demonstrated this phenomenon. So this was done at Joslin Diabetes Center at Harvard. And what you can see is here we have glucose, and here we have fructose. You notice glucose is in green, fructose is in red, for good reason. Glucose activates 2 enzymes that increase mitochondrial beta oxidation. It increases AMP kinase, the fuel gauge on the liver cell, to tell the liver to make more mitochondria. It also increases the activity of this enzyme down here, HADH, hydroxyacyl-CoA dehydrogenase, which helps the liver split those fatty acids into 2 carbon fragments so that they can be beta oxidized by the mitochondria. So glucose stimulates mitochondrial function. Fructose, on the other hand, does the opposite. It inhibits mitochondrial function, and it does it by inhibiting 3 separate enzymes. It inhibits AMP kinase by sitting in the active site and binding covalently to the active site and rendering it unavailable. It's an irreversible inhibition of AMP kinase. through methylglyoxal, one of the intermediate byproducts of fructose metabolism. In addition, remember that ACAD-L from Reye's syndrome? Well, fructose inhibits ACAD-L. When you inhibit ACAD-L, you inhibit beta oxidation. And lastly, fructose inhibits carnitine palmitoyl transferase 1A, which is the enzyme that regenerates carnitine that you need in order to be able to transport those fatty acids into the mitochondria for beta oxidation in the first place. So Ron Kahn himself, the CEO of Joslin, said about this, the most important takeaway of this study is that high fructose in the diet is bad. It's not bad because it's more calories, but because it has effects on liver metabolism to make it worse at burning fat. As a result, adding fructose to the diet makes the liver store more fat, and this is bad for the liver and bad for whole body metabolism. Exactly right. We at UCSF have looked at this question in a slightly different way. We have taken 43 children with metabolic syndrome and fatty liver disease. And what we did was we studied them, by the way, a Latino and African American, all high sugar consumers. We studied them on their home diet. And then for the next 10 days, we catered their meals, no added sugar. We took the added sugar out of their diet. We took their percent calories from sugar from 28% down to 10%. We gave them fruit, but no other sources of added sugar. Now, if you do that, you're going to lose 400 calories a day out of the diet. If you do that for 10 days, you might lose weight. We didn't want our patients to lose weight because if they lost weight, the critics would say, well, of course they got better, they lost weight. We wanted them to stay the same weight. So we had to replace the sugar with something equicaloric. We gave them starch. We gave them extra starch. In other words, we did a glucose for fructose exchange, isocaloric, in order to maintain their weight. And these are 3 of the 5 papers that we wrote about this. This just demonstrates the strategy. And what you can see here is that from day 0 to day 10, every aspect of their metabolic health got better. Everything got better. Here's their glucose tolerance test. Their glucose area under the curve went down 8%. Their insulin area under the curve went down 25%. And you can see here, they're starting to regain insulin sensitivity. We wanted to see whether the liver was making fat. So we gave them C13 labeled acetate to incorporate into new palmitate. This measures percent conversion of de novo lipogenesis. And you can see here, the fractional de novo lipogenesis got cut in half because of this maneuver with no change in calories and no change in weight. And so now let's look at the fat depots, okay, which we did with magnetic resonance spectroscopy. There's no change in subcutaneous fat because we didn't let them lose weight. The visceral fat went down about 7%, which is good. But take a look at what happened to the liver fat, down 22%. With no change in calories, no change in weight, just by removing the offending toxic substrate. So in cartoon form, here we have a fatty liver making triglyceride and VLDL, 9 days of isocaloric fructose restriction, fatty liver down 22%, DNL down 46%, VLDL down 49%, visceral fat down 7%, and most importantly, their pancreases started working properly. We reversed their metabolic syndrome with no change in calories and no change in weight, showing that this is not about obesity, it is not about calories. Rather, it is about the toxic substrate. And we're not the only ones who say this. This was corroborated by a group from UC San Diego and Emory University back in 2021, where they basically did the same thing and showed the same phenomenon. So mitochondrial dysfunction, de novo lipogenesis, is the first hit. The second hit, ROSs, reactive oxygen species, driving inflammation. So here we have a picture of 5 pictures of food, and they all have one thing in common. They're all brown. They're all brown, and the reason they're brown is because of the Maillard reaction, because the Maillard reaction is the glycation of proteins. The epsilon amino group of lysine binding to the aldehyde of glucose or the keto group of fructose. So the way to think of the Maillard reaction is the following. You can roast your meat for 1 hour at 375 degrees Fahrenheit, or you can roast your meat at 98.6 degrees Fahrenheit for 75 years. The answer is the same. We are all browning. And if you don't believe me, here is newborn rib cartilage nice and white, and here is 88-year-old rib cartilage nice and brown. And if you had orange juice for breakfast this morning, you are browning 7 times faster. Now, why does this happen? Here's the aldehyde group of glucose at position 1, binding to the epsilon amino group of lysine at position 1, forming a shift base, which then spontaneously decomposes to form this Maillard reaction, the Amadori product. This is, of course, how you get hemoglobin A1C. Well, this happens in all tissues, not just hemoglobin. Why does it happen? Well, here's why. Here's that aldehyde group of glucose at position 1. As you know, glucose can live in the linear form or in the ring form. Now, when it's in the ring form, that aldehyde is hidden. Now, this glucose molecule has 6 members of its carbon ring, and it also has a hydroxymethyl group up here axially. This is a stable compound. So at pH 7.4, 37 degrees, the glucose in your body is 99.2% in the ring form. Only 0.8% of it is in the linear form, where this aldehyde is available to engage in the Maillard reaction. Okay, and every time the Maillard reaction occurs, by the way, it throws off a reactive oxygen species, an oxygen radical. Fructose, on the other hand, is very different because fructose has its reactive component here at the position 2 group instead of the position 1 group. And so it can exist in the ring form also, but its ring form is a 5-membered ring, not 6-membered. And you'll notice that the hydroxymethyl groups are axial to each other. Allosteric inhibition, they drive it apart. Allosteric interference. Okay, so at pH 7.4, 37 degrees, 97% of the fructose is in the ring form, 3% is in the keto or the linear form, where this reactive keto group can attack an epsilon amino group of lysine. So fructose is inherently more capable of inducing the Maillard reaction. And every time you induce the Maillard reaction, whether it's glucose or whether it's fructose, doesn't matter, you will generate reactive oxygen species. You can see that in the dish. So here you can take a test tube and add bovine albumin to it and add fructose to it or glucose, either one, cap it with parafilm, put it in the window, and every day you come back and you measure it in your spectrophotometer and you can see the change in the fluorescence. The fructose is generating 7 times the rate of glycation with 100 times the rate of reactive oxygen species. So does this cause damage? And the answer is yes, in vulnerable liver cells. So this was work done by Richard Lee in vitro. What he did was he took primary hepatocytes in culture and he cultured them with either fructose or glucose or 2 aldehydes that are known to be toxic. And he measured the ED50. And you can see that fructose had no effect here, glucose had no effect here, okay, whereas the aldehydes did. But then what he did was he took those same hepatic liver cells and he treated them with a sublethal dose of hydrogen peroxide to render them vulnerable, to use up the antioxidants that were in the liver cells to start with. Then he added the fructose and the glucose. And what he saw was now the fructose, not the glucose, but the fructose, had the same ED50 that the aldehydes did. In other words, the only thing standing between fructose and liver damage is the degree of antioxidants. And so if you're antioxidant rich, you will only generate the fat. But if you're antioxidant poor, you will generate the inflammation as well, because he could prevent this phenomenon by the addition of anything that reduce the oxidation. So we know that fructose causes leaky gut because it nitrates tight junction proteins in the intestine, leading to translocation of bacteria and lipopolysaccharides. Those lipopolysaccharides will reach the liver through the portal vein, activate NADPH oxidase, generate reactive oxygen species. In a liver with low antioxidant capacity, it will lead to hepatocyte death and inflammation and fibrosis. So, second hit, glycation, oxidative stress, inability to quench. And now finally, the third hit, which is the part you probably have not yet heard about. And this is, we're very excited about it because it's our work. Defective packaging and transport. You have to get the fat out of the liver. So you may all be familiar with Jackie Marr. She is at San Francisco General Hospital and is very interested in NASH. And she uses a model called the methionine choline deficient diet, the MCD diet, not the McDonald's diet, but it might as well be. This is the fastest animal model to NASH that there is. And the reason is because methionine methionine, which is the second rarest amino acid in the diet, provides the methyl group to create glutathione. And glutathione, of course, is the scavenger molecule for those hydroxyl radicals, those oxygen radicals. So you need your glutathione. And if you don't have methionine, you can't make your glutathione. So if you're methionine deficient, that's a problem. And ultra-processed food is methionine deficient. In addition, she deprives the animals of choline. And choline, of course, is a component of phosphatidylcholine, which is necessary for ApoB100 export of fat out of the liver. So when you put the two together, you make a lot of liver fat, but it depends on the substrate. So what Michael Pickens, a pediatric GI fellow working with her, showed was that if you are sufficient and you give sucrose, it's the same as giving starch. No problem. But if you are deficient, starch looks fine. Sucrose causes all of this liver damage. And you can see the tunnel staining for the dead cells. So clearly, because of either inability to scavenge those reactive oxygen species, or because of the inability to export them out, you end up with severe fatty liver disease. We were interested in whether or not there was something else in the diet that might make a difference. So we looked at this question in adults, where we gave them C13 labeled acetate and C13 labeled glucose. C13 labeled glucose for this study. Okay. And what we saw was that long-term fat storage correlated inversely with the fat fraction, which you would expect. The more storage, the greater the fat fraction. Okay. There was an inverse relationship between the fat stored in the liver and the fat export. But when we looked at the individuals, turned out the individuals that were able to export the fat, we could see with their magnetic resonance spectroscopy that they had high levels in their liver of choline, okay, to make that phosphatidylcholine. Whereas the ones who had impaired fat transport, had long fat storage, they had low choline levels. Okay. We then looked at our kids and found a correlation between the liver fat fraction and the choline fat fraction. Sorry, the choline fraction. So the more choline, the less liver fat, the less choline, the more liver fat. So choline is a cofactor for transport of fat out of the liver. And these kids were choline deficient. Therefore, they couldn't transport it out. Therefore, they made more fat and it stayed in the liver. So we are posing to you the three-hit hypothesis of NAFLD. First, mitochondrial dysfunction and de novo lipogenesis, and you need a substrate for that. And the substrate is either fructose or alcohol, but in kids, it's fructose. ROS generation and inflammation, because fructose drives the Maillard reaction 7 times faster than glucose. And finally, malnutrition due to ultra-processed food, leading to defective packaging and transport because of choline inadequacy. So we have causation, not correlation, causation for sugar and 4 diseases: diabetes, cardiovascular disease, fatty liver disease, and tooth decay. We have correlation for sugar and cancer and sugar and dementia. We're working on those, and the data is really strong, but we still don't have proof. We're working on it. So what I would propose to you, the model of insulin resistance that you learned in medical school is wrong, that it's not the fat that drives the insulin resistance. Rather, it's the liver that drives the insulin resistance, that the fructose generates the fatty liver generates the insulin resistance, that generates the hyperinsulinemia, which then lays down fat because insulin working at the fat tissue drives adipose tissue accumulation. And therefore, you still have the same vicious cycle, but it started in the liver instead of in the fat tissue. This is the hepatocentric view. And then the muscle, of course, is along for the ride. Now, just so you don't think I'm fructocentric, there are actually 4 items in the diet that will all lead to the same liver phenomenon. Okay, it's not just fructose. Trans fats will do it. But we now know that trans fats are poison and they are coming out of our diet. Branched chain amino acids, leucine, isoleucine, valine. The work of Christopher Newgard at Duke has shown that leucine, isoleucine, and valine actually drive de novo lipogenesis as well. And that is 20% of what's in protein powder. So we have all these bodybuilders ingesting protein powder. This is not necessarily such a good idea. Alcohol, of course, and fructose. These are the 4 things that drive this fatty liver phenomenon. Now, what do these 4 things have in common? They have 3 things in common. The liver is the only site for energy metabolism. They're not insulin regulated, and there's no glycogen pop-off, and the mitochondria are overwhelmed. So we've tried to put together a unifying hypothesis of NAFLD. By the way, I don't like this MASLD at all, but we'll talk about that in a minute too. That fructose coming into the liver cell will generate that Maillard reaction, generating ROSs, will be metabolized in the mitochondria, generating ROSs, and inflammatory cytokines from the periphery will activate NADPH oxidase, generating ROSs. So you have this ROS pool in the liver that has to be quenched. And where do they go? They go to the peroxisome, because that's where the antioxidants are. That's where the glutathione is. That's where the vitamin E is. That's where you basically send ROSs to die. But if you don't have enough antioxidant capacity, then those ROSs will make their way to the endoplasmic reticulum, which are always right next to the mitochondria, and will engage in the unfolded protein response, or ER stress, endoplasmic reticulum stress, which can cause cell death or cell metabolic dysfunction. If you don't fold your insulin right, you have insulin deficiency. If you don't fold your insulin receptor right, you have insulin resistance. In addition, you're going to be making fatty acyl-CoA, and that fatty acyl-CoA could precipitate as a lipid droplet. And if so, now you've got an inability to generate an insulin signal through AKT because of the serine phosphorylation of IRS1. So fructose is not the only thing that causes insulin resistance, but is the thing we give to children. And it is the reason that children now have fatty liver. 5-year-olds have fatty liver. And there's no drug target. Okay, none of these newfangled NASH treatments are designed to handle this problem. The only option so they can reduce the inflammation, but they can't reduce the steatosis because only removal of the substrate can remove the steatosis. Only the resumption of normal mitochondrial function can reduce the steatosis. So the only options are to reduce substrate availability, that is, get rid of the sugar, reduce hepatic flux, that means fiber to reduce how fast the fructose reaches the liver, improve transport, that's choline, and increase clearance, that's exercise. So it is diet and exercise, but for very specific reasons and purposes, and it's not just any old diet. It's a real food diet. There are no medicines for any of these problems. And then finally, the third myth that we must talk about. Weight loss is the treatment for fatty liver disease. So this is from the American Association of Clinical Endocrinology Clinical Practice Guideline for NAFLD. You all remember it. You all, I'm sure, have studied it. Maybe some of you are even on this. I don't know. I'm just going to show you one sentence. While no US FDA-approved medications to treat NAFLD are currently available, management can induce lifestyle changes that promote an energy deficit leading to weight loss. We have shown you this has nothing to do with weight or weight loss. What it has to do with is the generation of the fat in the liver in the first place. Okay, that's the problem. So really, it shouldn't be MASLD. It should be DAFLD, diet-associated fatty liver disease. And really, to be honest with you, for kids, it should be FAFLD, fructose-associated fatty liver disease. So Hippocrates famously said, let thy food be thy medicine. Yeah, but Hippocrates never met McDonald's. And if he had, he probably would have said something else. He probably would have said, good food is medicine, bad food needs medicine. And fatty liver disease is the perfect example of how bad food needs medicine. So with that, I want to thank my collaborators at UCSF, at Touro University, at Children's Oakland, at San Francisco General, at Stanford, and at Children's National Medical Center, who all contributed to the studies I've shown you and put this all together. So with that, I'm happy to answer any questions, and I hope this was helpful and entertaining.
Thank you very much, Professor Lustig, for your presentation. That was, from my point of view, very provocative and stimulating to think the things in another way.